Alright, so next we're going to go over monohybrid crosses and how to actually do problems like that. So here we have a parental mono, P1 stands for the parental generation monohybrid cross. Your trait that you're looking at, seed shape, capital R is round, little r is wrinkled, and you are crossing round seeds with wrinkled seeds. So you have two capital R's and then two little r's. How do I know from the setup of this problem that I'm going to be using those specific genotypes? Well, the clue in this slide is going to be P1, or the parental generation, which means you're using those pure plants and you're crossing them. So that's how you know that it's referring to something that has the same genotype, or a homozygous genotype of big R, and a homozygous genotype of little r. So as you can see in the bottom right here of the screen, there are 4 squares, and we are going to fill those out. So on one side we have the big R from one of the parents, and then at the top you put the little r from the other parent. And then you combine them together in the box, and you should get something like this. As you can see, the letter just drops down into the boxes below it or across from it. So all of these genotypes happen to be the same. They're big R, little r, which is heterozygous because you have a combination of the dominant and recessive gene. But while the genotype is going to be big R, little r, what you're actually going to see is that they're all round seeds. So the phenotype is also the same. You're going to have the same phenotype and the same genotype for each of the offspring of this generation. So this is your F1 generation. So you have homozygous dominant versus homozygous recessive, and that's because it's coming from that parental generation. So the P1 generation. And then all of the offspring, you'll notice, were heterozygous or hybrids, and they are the F1 generation. So just like in the previous video when it talked about going from the P1 to the F1 to the F2, those are just generations. Alright, so then let's look at the F1. Now we're doing an F1 monohybrid cross. So if you think about it, the F1 generation, you notice that all of them were the same, all were alike. So this tells you that the parents are going to have the genotype big R, little r, because it's the next generation. You're crossing the generation that you just found out about in the previous problem. So you're still looking at seed-shaped, but this time, and you're crossing 2 round-seeded plants this time. But since they are the F1 generation, they are going to be heterozygous. So the genotypes you are crossing are the big R, little r. So you put those on the sides of your boxes, and then you're going to fill in the boxes according to which letter. So the big R on top drops down to these 2 boxes, and then this big R on the side goes over. And you end up with big R, big R, big R, little r, big R, little r, and little r. So you have a homozygous dominant with the big R, a homozygous recessive with the little r, and a heterozygous big R, little r. So you have 3 different genotypes in this Punnett square, and you actually have 2 different phenotypes too. You're going to have both round and wrinkled seeds because you have that recessive trait. So your ratio is going to be 3:1 for your phenotype because big R, little r is going to be round, and so is big R. And those are 3 of your 4 squares. So it's a 3:1 phenotype ratio. But if you look at the genotype, you have 1 big R, 2 big R, little r's, and then 1 little r. So your genotype ratio for that is 1 to 2 to 1. Okay. So if you cross a heterozygous and a heterozygous, you are going to get 25% of them being homozygous dominant, so 1/4. So one of the boxes was homozygous dominant, big R, big R. And then 2 of them were heterozygous, big R, little r, so that's 50% because there's only 4 boxes. And then 25% was homozygous recessive, which is little r, little r. And this offspring with the 3-to-1 phenotype ratio is called the F2 generation, which is a vocab term you should remember from the previous video. And the genotype ratio is 1 to 2 to 1. Okay, so that is the basics of your monohybrid cross. And please rewatch the video as much as you need to get a grasp of this concept, because I'm leaving you some problems that you are going to need to solve using the method described in this video.